Hello everyone. We are going to talk about a very important organelle which is seen in the cells of aerobic organisms, and this is a mitochondrion. Mitochondrion is the singular name that we use, and if many mitochondrion are present, we call it as a mitochondria in general. So let's talk about more of mitochondria in detail. Mitochondria, and as I told you, the singular is known as mitochondrion, is a cylindrical shape, or it can be a sausage shaped structure. As you can see over here, this is the organelle present. It looks sausage shaped in structure. Then they are double membrane bound structures, organelles, which are present in the cytoplasm. Mitochondria was first discovered by Colecax in 1850, and they were named so by Benda in 1897. Mitochondria, until and unless we don't specifically stain them, they are not easily visible under the microscope. If we look at the mitochondria, we see that each mitochondrion is around 0.2 to 1 micron meters in diameter and around 1 to 4.1 micron meters in length. Then, the number of mitochondria that are present in a particular cell, it actually depends upon how much physiologically active a particular cell is. So, it would be correct to say that higher is the metabolic activity of a particular cell, higher will be its number in a particular cell. So, higher is the physiological activity of a particular cell or the metabolic activity of a particular cell, higher will be the number of mitochondria present in that particular cell. For example, if we see the germinating seeds, they are highly metabolically active cells. So, in them the number of mitochondria is comparatively much more as compared to a normal cell. Then, mitochondria, these structures, they are absent in prokaryotic organisms, obviously, because these are membrane bound organelles, and membrane bound organelles are not seen in prokaryotic cells. So, mitochondria will not be present in any prokaryotic cell, as well as they are absent in anaerobic eukaryotes. But these structures, these organelles, they are present in all aerobic eukaryotes except for mature RBCs, which obviously do not have many other cell organelles because of their functional roles, as well as the mitochondria are not present in sieve tubes of phloem as well. Then, the mesosomes that we see in the case of prokaryotes, we have also studied about them when we discussed the prokaryotic cell. So, mesosomes are almost the same structures, the analogous structures of mitochondria which are present in prokaryotes, because they both perform almost the same type of a function. So, we can say mesosomes of prokaryotes as well as the mitochondria in eukaryotes, they are analogous structures performing the same function. So, this was some of the introduction of mitochondria. Now, let's look about the structure of mitochondria in detail. So, this is a picture of a mitochondria. This is the whole structure. We can see over here in the mitochondria, each mitochondrion is bound by two unit membranes. So, we can see over here, this is the outer membrane present, this whole. There are two membranes in mitochondria. So, this is the outermost membrane, and this is the inner membrane which is present in mitochondria. So, mitochondria are double membraned structures. Now, the area between the outer membrane as well as the inner membrane, this area is known as the intermembrane space, or it can also be known as the perimitochondrial space. So, the area which is present in between both the membranes refers to the perimitochondrial space. Then, the outer as well as the inner membrane, they divide the whole mitochondrial lumen into two chambers. One will be the outer chamber and the other is the inner chamber. So we can see over here, this area which is bound by the outer as well as the inner membrane, this will be referred to as the outer chamber. And the area which is limited only by the inner membrane, this whole area present inside, this is known as the inner chamber. The inner chamber or the inner compartment, as we can also call it, it is filled by a dense homogeneous substance which is also known as the matrix or the ground substance. So inside the area which is limited by the inner membrane, this whole area which is also known as the inner chamber, it is filled by a homogeneous substance that is known as matrix or the ground substance. The outer membrane is quite permeable, that means it allows most of the things to enter inside because of the presence of special proteinaceous substances known as porins. Whereas the inner membrane is quite regulative, the inner membrane is selectively permeable, and the inner membrane, if we look very carefully, it is folded inwards towards the matrix. So can you see these infoldings, these ones of the inner membrane, this as well as this, then these all infoldings are known as cristae. These infoldings of the inner membrane, they help these cristae, which are singularly also known as crista, they help in increasing the surface area for the performing function of mitochondria. So this is the arrangement in which the membranes of the mitochondria are arranged. Now, so let's talk more about cristae in detail. If you look at cristae over here very carefully, you will be able to see these small particles which are attached to cristae. These small particles which are attached to cristae are known as oxysomes, or they are also known as elementary particles, or they are also known as Fernandez-Morin particles, or simply also known as F0, F1 particles. So we can have a look over here. All these structures which are attached to the cristae, these are actually known as the oxysomes. And you can also see this is a expanded picture of the oxysomes. They look these ways, and that is the reason they are also known as stalked bodies, because they are having stalk present in them. This is another picture of the F0, F1 particles, an expanded picture of those. We can see this is the inner membrane. This is the infolding of the inner membrane, which is known as the cristae. And to the cristae are attached these structures which are known as the F0, F1 particles, also known as the oxysomes. So let's study more about these oxysomes in detail. So each of these elementary particles is made up of a base, a stalk as well as a head, and is around 10 nanometers in length. So we can see over here, this is the base of the particle. This is the pedicel, also known as the stalk of the particle, and this is the head of the F0, F1 particles. The F1 particles are associated with ATP production during aerobic respiration, as they contain the enzyme known as ATP synthetase. So ATP formation takes place over here in the F0, F1 particles, and this portion forms the F1 particle, whereas this portion forms the F0 particle. And it is here in the F1 that the enzyme for ATP synthesis, that is ATP synthetase, is present in it, which helps in the production of ATP. Mitochondria are therefore, because they are associated with the formation of energy, because they are associated with the formation of ATP, that is the reason mitochondria are also known as powerhouses of the cell. So, Apart from these oxysomes, the mitochondrial matrix, it also has single circular DNA molecule which is having a high guanosine to cytosine ratio, and the matrix also has few RNA molecules. It has 70 S ribosomes, as well as it also has some of the other components which are required for the formation of proteins. So, because a mitochondria matrix has all these structures, that is the reason the mitochondria are also known as semi-autonomous organelles. Why? Because they can form some of their required proteins on their own. So, that is the reason they are known as semi-autonomous. Now, why they are known as semi-autonomous? Because they are able to form some of the proteins and not all of the proteins that are required. So, for all the other proteins, they have to depend upon the cytoplasm and the other machinery of the cell. So, that is the reason they are known as semi-autonomous. That means they can form some proteins themselves, but not all the proteins that are required for their survival. So, now let's talk about the functions of mitochondria. So, as we have seen, mitochondria help in the formation of ATP, that is the energy currency of the cell. Then, the intermediate products of cellular respiration that are used for the formation of steroids, for the formation of cytochromes, for the formation of chlorophylls, etc., they all are also formed in the mitochondria. Then, the biosynthesis of some amino acids, that is also done here in the mitochondria. Similarly, the calcium ion concentration is regulated inside the cytoplasm by the mitochondria itself. It is also the site of thermogenesis. That means it produces energy, it produces heat. So, it is the site of thermogenesis as well. And obviously, why? Because it releases energy during the process of respiration. Mitochondria, they also contain the electron transport system, all of which is required for the formation of ATP. So, these are some of the functions of mitochondria, which as an organelle is very important, and one thing, it is also known as a semi-autonomous organelle. Do you know there is also another organelle which is known as a semi-autonomous organelle? Just think about it, and then we are going to discuss this organelle in the next video.
To buy these books, you can go to www.cengage.co.in, or you can also find the links in the video description given below. Thank you.